Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology. Then I was a fact checker and editor for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC dedicated aimed at the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist. I'm currently working for a scientific journal.
Every other week, we meet to talk about microbiology, and on a typical day like today, we do an overview of some of the coolest microbiology papers that we've seen in the last 2 weeks.
Yeah, and every now and again we try to do a deep dive. So if you've got an idea for a paper you want us to take a deep dive into, message us at microtwjc at any one of the venues listed in the doobly-doo, and let us know if something's caught your fancy.
Yep, you can follow along with the papers that we discuss on either week in our shared Zotero library, linked in the doobly-doo below. Our social media handles are there, and yeah, hello, Cordo, in the chat.
Hello. And we want to hear from you, so please use the comments or tweet us at microtwc. So what kind of papers do we have today, Danny?
All right, so I mean, we skipped our last week, and we've been out for like a month. So these papers have been accumulated not over 2 weeks, but over like essentially a month. But hopefully they're super exciting. I mean, we're excited to talk about them. We got something about surviving phage infection by ditching their cell walls, testing an mRNA vaccine for bacterial infections, plasmids that hijack bacterial regulatory networks to facilitate their spread, symbiosis between fungi and algae, just shy of lichenization, and bacterial gas ballast tanks.
Yeah, and we also have some how long bacteriophage tails get made. We also have using unicellular algae to help create solar panels, apparently. And all that and more. So I think we can get started on the first paper, which is let me cue this up. A single-dose F1-based MRNA-LNP vaccine provides protection against the lethal plague bacterium.
Mm-hmm.
Not the Sorry, plague. I am under the weather at the moment, so it's not COVID, but it is an annoying cold, and I've got a slight fever. So don't worry, I will make it to the end of this.
We appreciate you having me here. Yeah, this F1 based vaccine. So mRNA vaccines, these are famous now because sort of the two high profile first vaccines to make it to market for COVID-19 were based on this technology. Essentially taking a strand of mRNA, which has the coding instructions for some sort of protein typically, you put that inside of a ball of lipids, and then you inject that into people. Those lipids fuse to whatever cells. I think this is kind of still like an open question. What cells exactly do the LNPs fuse to? But once they fuse into those cells, they release the mRNA, and then those cells end up making what's being programmed in there. And typically, for vaccine purposes, they're going to program in some sort of antigen that we want to create an immune response towards.
Yeah, it's interesting because we haven't really seen this used for bacteria. We've generally seen it used for viruses, because I think the way these vaccines work, they almost mimic the way that viruses work, in that they inject the genetic material is injected into the cells, and cells make the antigens, and so it's odd to see it used for bacteria, especially with bacteria that's generally extracellular, as I understand it.
For sure, yeah, like it's sort of very understandable, like when we think about vaccinating against viruses using these LNP vaccines, because they're recapitulating like one of the ways in which your cell would naturally see these antigens, which is a cell gets infected and starts producing viral proteins. Those proteins get processed somehow and displayed on the surface of that cell, or even they just migrate to the surface of the cell. But it's not really like 100%. There's no specificity to these LNPs. They fuse to whatever they happen to come across. And so for that reason, it's also like not 100% known, I would say, like what is the most important cell type, right, that takes up this mRNA. that
Mm.
participates within immunity. I mean, we can make some good guesses based on what we know of the immune system, but probably for different diseases, there's going to be a different target. And unfortunately, one of the things that I found unfortunate about this paper is it didn't really dive into that information. Mostly, it's just trying to adapt this current scientific platform for a plague vaccine. So in my mind, it was kind of like a fusion of like, oh, aren't we afraid of the plague? And like, isn't this a cool technology? Just to try to see, just to show people that, you know, the latest thing worked on it.
Yeah.
So let's see, what figure do we have up here? This is, oh, they did it in this is the mouse survival, or this is in cell culture.
Yeah, this is, I believe, the mice survival. So they use lethal plague challenge to test this against the RF1 plus alum kind of. That's their positive control, and they've got their SP-CP-CAF1, which is their mRNA candidate, if I understand it correctly.
Yeah, and so it's making the F1 antigen, which is, I think Fuzz said, it's like a outer pilus type thing on the surface, or capsule. It's a capsule of the Yersinia pestis. So just like some extracellular protein stuff that usually prohibits phagocytosis, I think is one of the ways in which it works. And so the idea is if you can make antibodies towards it, then instead of preventing phagocytosis, it kind of drives more cells to actually go and grab these things.
Yeah, the thing that really stands out to me is how the titers for the antibody titers for the mRNA are very low compared to like the positive control, and I feel like that's a big thing they try to address during this paper, because
essentially they have to do a bunch of work to make sure that it's even equivalent to the to the way in which regular sort of protein-based vaccines are working.
Yeah, and I think that's like kind of indicative of how this almost seems like the wrong strategy to use to try and vaccinate against Yersinia pestis, but they're trying to make it as better. They've already handicapped themselves, and then they're kind of trying to. That's kind of my impression of it.
Yeah, no, absolutely. I mean, again, like you could see this as like an attempt to somehow flesh out how this platform could be used in different ways, but as you say, like you could just, if for me when I read this paper, I like kind of zoom out one level and I'm like, why did they choose to use this approach in the first place when we know it's going to be hobbled in certain ways? At the end of it, but what I do find interesting is that they do end up taking away some lessons from this, like that like they tried to put a secretion tag onto this protein in the hopes that it would get secreted, but actually that secretion tag didn't really work out that well for them, and they should just remove the secretion tag and it worked better. But it's like I find that interesting, right? but at the same time, I'm like, is that going to be a translatable discovery to like other contexts, or is this just like empirical troubleshooting on an approach that I'm a little bit doubtful of in the first place, anyways?
Yeah, I mean, I do think that it's interesting, like, because I feel like with mRNA vaccines, there might be like a quicker route to them being improved because the actual products themselves are like the products themselves are very similar across them. They're all just bits of RNA, and I'm not sure like whether the regulatory systems have caught up to whether like what they translate is an important factor in that or not. So it might be like the way to take advantage of all the infrastructure already in place for making the SARS-CoV-2 vaccines and using that for other diseases. I think that might be one reason to use mRNA vaccines, even for things where they might not be the best choice, because there is all that infrastructure in place to make them now.
Right. Yeah. So like technologically, it's kind of fascinating. The market is kind of primed to find that fascinating. But like from our scientific point of view and looking at this, it's sort of like it's a bit more doubtful from that outlook.
Yeah. I mean,
but nevertheless, they are able to make a bunch of modifications. It does seem to be a not bad vaccine by the end of their optimization protocols.
Yeah, I think that's an impressive like feat in itself, and if they do get it to work, then it does improve the idea of like for other like diseases. Perhaps it could be
Mhm.
a nice, interesting test for because at the moment we're focusing on mRNA for like maybe cancer vaccines, viral vaccines, but then there are lots of bacteria that out there that could potentially be this could be a useful thing for. So I think doing this kind of experiment could be useful, but I do feel like from the get-go there is that handicap there.
Yeah, and that logic that you described just a few moments ago of saying like, oh, but we're just taking advantage of this moment where there's a lot of infrastructure, regulatory maybe hasn't fully caught up to this idea of like making sure that the produced object is also vetted. Like to me, that's a temporary thing, right? Like eventually we want to live in a world where there are the right like regulatory
Yeah,
controls
that's
over that
right.
type of stuff. So it's like that opportunity is fleeting in my mind. And if one of the ways you find this paper exciting is that I would just ask to reconsider and maybe think about like how like we want to just develop a strong basis for making. We want to choose the right tool for the job in all cases, and it doesn't really matter like where the regulatory structure is. And I mean, it does matter in the practical sense because you're not going to be able to manufacture this or sell this or whatever. But yeah, I don't know. Choosing the right tool for the job, I think is like that's my takeaway from this paper.
Yeah. I think we've said everything we need to on this paper, so we can move on to the next one, which is chemokines expressed by engineered bacteria recruit and orchestrate anti-tumor immunity. So this is an interesting one because we have talked a bit about how bacteria and tumors can synergize sometimes. Tumors have their own um, microbiome.
Microbiota.
Yeah.
And then I think we've also talked about the fact that people have seen this phenomena where circulating bacteria accumulate sometimes in tumors. So there's also some weird targeting phenomena that happens between bacteria and tumors.
Yeah, and so this paper almost uses that as a way to test out whether this specific chemokine would be a good treatment for tumor cells, because half of this paper is about the bacteria, half is about like looking at this chemokine called CXCL 16, and which is a chemokine that is used to, I think to, I mean, it's used
attract macrophages, something like that.
Yeah,
Attract
that's right.
lymphocytes. Yeah.
And so, in order to like deliver this tumor, they created this gene circuit called the synchronized lysis circuit, where essentially once the bacteria go into the tumor, they have all the CXL16, and then they kind of pop open and release it from the tumor, and then that's supposed to attract all the T cells and other anti-tumor responses to the tumor cell cell.
Yeah,
So
absolutely. Yeah, so these chemokines, they're made by our immune system in order to, signal where immune cells should go. And essentially, the authors here are trying to deliver these chemokines artificially into tumors. And yeah, they're exploiting this idea of bacteria making it to the tumors and their lysis circuit, which essentially treats bacteria as if they were just bags of delivery bags, right? And like they can induce them at any moment to pop open and release their payload.
Yeah, and we see that they get some interesting results on like getting the tumors to grow less fast, and also like we see some things about T cell infiltration, and then they move on to not just looking at CXCL16, but also other potential chemokines to add into it to perhaps synergize that. So instead of not just recruiting T cells, but also recruiting dendritic cells into there, so they get a CCL20 also be created by these bacterial bombs. Is what my sickness brain is going to. These bacterial bombs that go into the tumor cell and pop to create a cocktail of chemokines that can attract the immune system to them.
Right.
I think this is kind of an interesting thing, not just because as a treatment, they have to give these mice like regular injections daily, but also I think this is an interesting test bed to see how chemokines can work in a certain situation. So I think that's basically the fascinating part that I'm looking at with this.
Yeah, I think it's interesting. It's one of those things where I'm always surprised that bacteria are a good chassis for this type of thing, because you can imagine bacteria floating through your circulatory system and lysing, it's releasing all of the outer components of the bacteria as well into your bloodstream. So like, but they're using Nissl, which we've talked about before. It's like probiotic E. coli. And I guess there's I guess the lipoticoic acid, like the outer cell wall and stuff, isn't as immunogenic as it could be. It's still a really weird chassis for me to think about. Like, probably more of a model chassis within mice than a chassis that we would see used inside of people.
Well, yeah, that's the paradox, right? Because if you're going to have a probiotic bacteria that goes inside your system, you don't want it to be immunogenic, because otherwise your system will react to it massively.
Mm-hmm.
But also, part of the thing that would make it good for tumors is if it was slightly immunogenic. So that's kind of the balance. Because again, this is something that, because I know that like So for immunotherapy, one of the earliest versions of it was where like people were infected by live bacteria and it migrated to tumors, and that created an immunogenic response.
Mm-hmm.
But that has so many other dangers and safety issues associated with it, that having a less immunogenic bacteria, but maybe has something that is immunogenic that activates when it's in the tumor, that might be a useful thing to have control over.
Yeah. So I mean, in this way, I think I just want to highlight like how experimental this treatment really is.
Yeah.
They even say like I think what you said, Fuzz, earlier, like how they're framing this as they're really learning a lot about the chemokines and how the chemokines induce movement of cells. Whether or not this particular modality is going to be a therapeutic modality, to me, that's a little bit more questionable.
Yeah, I agree with that. I think this is very early to look at it as a therapeutic modality, but I think it's still quite an interesting test bed to test how chemokines interact in an in vivo situation. So
yeah. And also, I'll say it's kind of wild to think about bacteria making human signaling molecules. That's kind of just an interesting framing. And again, I think for people that are very familiar with genetic engineering, like we know E. coli is the workhorse to make every protein we need. So thinking about what if they made those proteins in different environments, that's kind of a really curious thought.
Yeah. And like I'm thinking, like, just because we only have bacteria that create insulin, but then if you can tap into like the microbiota to have something like have a bacteria that creates insulin in response to certain things, and I mean, I'm going wildly off track with this, but
yeah. Okay, so let's move on to the next one.
Yep.
Our next paper is Lform conversion in gram-positive bacteria enables escape from phage infection. So Lforms are this type of bacterial morphology that's been observed in the past, particularly when cells have been exposed to antibiotics. Like a lot of antibiotics target something in the cell wall. Many of them target something in the cell wall. And so it was observed that you can find persister cells that grow in the presence of antibiotics. And when they looked at those persister cells, they're like, oh, they strangely don't have cell walls anymore. And so for a long time, the way that these bacteria were called Lform bacteria. And the main thought was that they persisted after some sort of antibiotic treatment. The switch, like it's a big question. Like, how do they switch between these formats? Like, what's the real role in ecology or whatever of this particular format? And in this paper, because the logic is that these Lforms occur when something's happening to the peptidoglycan, to their cell wall, and phages infect typically through the peptidoglycan. They have to lyse it, they have to bind to it. They want to just follow up and say, well, does it mean maybe that phages can induce this Lform? And then that would give us like another way of thinking about how the Lform is useful for the lives of these bacteria. And so they treat a bunch of bacteria with phages, and the key is that they they're in an osmoprotective environment, because you can imagine if you lose your cell wall and you're just a cell membrane, now you're susceptible to having some osmotic stress happen, like water could flow into your cell and burst you. So you have to have like the right balance of salts on the outside in order for these Lforms to survive. So that's one caveat to this, or like something we should remember as we're looking at these figures. They're doing this in osmoprotective scenarios, but we'll see at the end of the paper, there are some osmoprotective scenarios in our bodies where this could be useful. It's urine, is the spoiler.
Yeah, this is interesting because I've heard about Lforms for so often, but it's hard. But usually, it's generally like been cast as, oh, this is a weird thing that bacteria do when they, and not like as an evolutionary protection that from phages. Whereas this one kind of builds up that story a lot more and makes it make a lot more sense in my mind as to where Lforms sit in the bacteria like kind of lifestyle.
Yeah, absolutely. Yeah, so I mean, I think you just scrolled past a picture where you could see, like, in the non-osmoprotected scenario, like, they do lyse.
Yeah.
Yeah, so, like, this is only relevant in some subset of environments that these bacteria find themselves in. And it's also the trigger of this is the endolysin activity. So, like, you don't have to infect the phages. You don't have to use whole phages on Listeria to make this happen. They were also able to just use like the lytic proteins from the phages onto the bacteria, and then these Lforms appear as well, which I will point out is a really interesting observation from the standpoint of people thinking about using phage lysins as therapeutics. Right? Like, so it means that in some set of conditions, like bacteria can move on from that challenge and just persist in another form. So like, right, those are not the sort of silver bullet. As I mean, I don't think anyone truly believes that like new therapeutics are a silver bullet, but like just, you know, to keep in mind that bacteria may be able to survive that as well. Oh, in the next figure, in figure 4, they have like a great like EM tomograms, and you can see like they are able to capture these snapshots of how the small break in the cell wall creates like a little opening that the inner membrane can just sort of bleb out of. And it's really, they say it's just driven by pressure, that like the inner pressure of the cytoplasm fills up a membrane bleb, and then maybe there's a mechanism that cuts it off, right? Like, who knows, right? What's going on in this sense? They only have the images to sort of guide what's going on.
But that's another interesting part, because it's almost like stemmed out of the study of like the membrane vesicles that the bacteria can create in some when they're stressed out. Right?
Yeah. I mean, maybe that'd be really interesting. Like maybe some of the machinery that's used to make those membrane vesicles is also important in this context, because you can imagine there's a very specific sort of morphology of the of the thin neck where you might want to, there might be machinery that like pinches up properly so it doesn't just like pop open and break.
Because again, this almost reminds me of that paper where like we looked at like bacteria on an antibiotic gradient, and the bacteria were running towards the gradient as if it was like a competing bacteria, and they were like trying to destroy them. Like when it's under stress for an antibiotic, it releases outer membrane vesicles that can be targeted to destroy the. Again, I feel like I pulled us very off topic again. I'm sorry.
That's fine. I'll say though that the difference between like outer membrane vesicles and what they're finding here is that these, once they put them back into like a regular environment without endolysins and stuff, they'll become their walled forms again. So there's this very much sense that like whatever is getting butted off into these bags, like it has like the genomes in there, right? Like everything's there so that they can reproduce again. And it's like, yeah, so it's like they're really just escaping their cell wall through a tiny hole.
Yeah.
to live again, to replicate again another day when conditions change in their favor. And maybe the last thing that I want to say, so, you know, the final figure of this paper, figure 6, like they tried in urine. Because like the idea is that urine is osmoprotective. And so like the fact that they can find, you know, this phage-induced Lforms in urine would mean that like like Lforms live in urine essentially, can live in urine. So like it's not so strange to think of them persisting. Like for example, in a UTI infection, right? You could have not phage-induced but antibiotic-induced Lforms in that particular environment. And then there's just one observation that I wanted to pull out from this paper that I thought was interesting is uptake of plasmids. Yeah, actually, that's a really good point. With gram positives, this is actually a common. There is this spheroplast sort of style of transformation, right, where you want to digest the cell wall away first and then shock in plasmids. So yeah, it's already used, I'll say. Like removing the cell wall is already used. This could be maybe a different preparatory technique if you think about it.
Yeah,
like a low cost. I mean, endolysins, I feel like we haven't really used them as a lab reagent in the past. Like we're exploring them as a therapeutic reagent, and it's kind of interesting to think they could be a lab reagent as well, digesting away the cell wall in order to have just a membrane bag that you could more easily shock something across, let's say, or chemically induce something across. You're right. The final observation that I wanted to share with people was that it's not the bacteria. They do a test where they like have some glowing bacteria and some not glowing bacteria, and the phage also carries with it like a reporter gene, so they can see like where does the Lform occur? Does it occur in just the bacteria that are sitting around the infection, or are they the actual organisms that get infected are the ones that produce the Lforms? And they find that it's the bystanders actually. Oh. So it's not the cells that get infected by the phage that are making the Lforms. It's just the cells that are around that are, some endolysins reach their cell wall, digest it away, and then they kind of escape infection by the phage by ditching their by blebbing out, and then they don't have any receptors, right? The phage need the cell wall in order to find these bacteria, and so without it, they're able to like wait out the infection course in this format. So then when things again go back to normal, they can replicate again, reform their cell wall, and be protected from osmolarity again and go on their daily way.
Yeah, I think that covers that paper. I can move on to the next one.
Yep, let's go on to the next one.
Is multicopy suppressor screens reveal convergent evolution of single gene lysis proteins. So again, this is still on the topic of phage lysis, and this paper aims to try and like understand how single gene lysins work. So some like phages in the Viridae phages, they cause host cell lysis with just one gene, and the issue is like, even though these,
I think that I just want to clarify. So like SGL, it sounds like that's just like a broader form for endolysins, maybe.
yeah. I think so. It is. So I think like one of the first phages that was ever like sequenced was MS2, which had one single gene lysins, and the people are quite interested in this because of the possibility for using phages as like phages in therapy, and that's how I came across this paper. It was sold to me as, look, they found a way to find hundreds and thousands of how phage lysins work, but with the paper itself, it uses some like forward genetics to try and understand how these lysins work, because a lot of the issues is like some of them, it's quite hard to figure out what they're actually doing to the cell wall. And
well, because cell wall is like important for survival. So like one of the typical ways that bacterial geneticists would go around figuring out a problem is they would make a mutant library where every mutant in the library had a mutation in one of the genes. And then if something doesn't work on that library, then they're like, oh, it must be this gene that's important to mediate the response. But you can imagine that if all the peptidoglycan cell wall genes, they're important for cell survival, then you're not going to see those members in your library. And so you have to choose a different way of approaching screening for phenotypes that interact with the cell wall. You can't do it in that sort of negative, reverse genetic style.
And I'm funny you should say that. They do create a library of these mutants using with DNA barcodes attached to them, and they try to screen out which ones are the ones which, when we delete them, which ones are the ones that don't work with the single gene license, which
Right.
is
Well,
a good
no,
way to find out.
Wait, they do it in the forward way first?
Yeah, I think, forward.
they're like overexpressing one of the genes.
Yeah, that's
Right?
right.
Like this is why like you can't do it in the reverse way. You can't like make a library and figure out which one broke. You have to instead do it in the forward motion where they're saying,
Yeah.
let's make more of something. And maybe if they made more of it, then it'll make it resistant to being, I don't know, like it'll figure a deficiency, or it would like have them lyse better or something like that.
Yeah, I think you're exactly right. I think I kind of yeah. So they did that to try and and I think they unveiled there's a couple of genes that they think are involved, but they have to screen out other things that. So what is protect? Oh gosh, sorry, I've lost my thread completely. On this one.
Yeah, I mean, no, I think that's, to me, that's the big takeaway, because it's right. They had to produce this forward screen, so then they pull out gene candidates that that do allow the lysis of these, or that when they make more of it, it overcomes the ability of these license to break up the cells, and that's giving them insight onto what genes these license are working on. Because the licenses are expressed, they have a panel of license, and they're expressed from plasmids that are inside of this big library.
Yeah, and I think the issue is trying to work out which ones are the ones being directly interacted with by the license, and which ones are compensatory to the effects that the license produce. So sometimes if you overexpress a gene in the same presence of a license, it might not just be that the license is acting on that; it might be compensating for the damage that the license is doing.
Exactly.
Yeah, my apologies. I lost the ability to read my own handwriting on my notes. So
no problem. Yeah, I think that's, to me, that's an interesting takeaway. Like, it's a way of learning. Like, this is one of the well, first of all, the other thing is that they're using deep sequencing to interrogate their library. This is like the new way now. Like, before, people would be looking at colonies on plates, picking colonies, doing genome preps from each one, and having to screen through a lot. Now you can just do it in a big flask, essentially. Your library is just in a flask, and you sequence the whole flask, and you see where the abundances change. So yeah, this is just a very modern approach to bacterial genetic screens, and in taking this approach, they're able to learn something about the license and what genes they target, which might be good for design, might be good to find license that, we're really just learning a lot about this class of molecules. And as I said before, there are common, and Fozzie said it too, it's like a therapeutic thing people are interested in.
Yeah, there are 10,000s of genes that are like this, and the question is working out which one is the right one and how bacteria might adapt to them, because essentially when you're creating a phage cocktail, you want to make complementary ones, and trying to figure out ones that hit different parts of the bacterial cell wall construction steps is the thing that we want to do.
Yeah, because that would be like using different, right? That would be like using the antibiotic cocktail. So if they got resistance to one, they wouldn't have resistance to the other. So that means resistance as a whole will hopefully not evolve as easily, because we're using these combination therapies and making the evolutionary landscape that bacteria have to move across in order to survive much more difficult to navigate.
Yeah.
All right.
Okay.
So
next paper. Is
still phage related.
Still phages. Conformational dynamic control assembly of an extremely long bacteriophage tail tube. So this focuses on this hyperthermophilic phage that lives in these hot springs, and it's called P7426, and it has a very long phage tail. So it's 1 micrometer, which in virus terms is quite a lot. And tails do some important things. They recognize a host, and they regulate viral attachment. And so eventually, these tails consist of a tape measure protein and a tail tube protein. So tape measure protein is supposed to initiate the tail assembly, and the tail tube protein just forms the majority of the tail tube architecture. And so this kind of lives in a temperature of about 65 degrees C. So it's very extreme conditions. And so we'd imagine that the tail tube is quite stable. And this paper looks into that structure.
It's kind of a mystery, right? This paper is saying, like, how can a phage that lives in such a warm environment where, in warm environments, everything's bashing against each other. So like things have to be very stable in that environment. So how can a phage live in that environment and also have this ridiculously long tail, which I think in figure 1A, like I didn't realize, I didn't even think regular lambda tails were that long. Like, I don't know, I guess in my mind when I draw them, like a child with crayon, like I draw like the head approximately the same length as the tail. But that's not true for lambda, and that's certainly not true for the P74s to 26s.
Yeah, it's a long noodle of a virus.
Right. And so, the mystery is how does it fold into such a nice tube when the temperature's so, right? What extra features does it have to make stability?
Yeah, and this paper uses Cryo-EM to look at the tail structure, and they kind of corroborate that with like molecular modeling to essentially like build up this idea of how it's constructed and how it's been stabilized. So you get some nice like Cryo-EM, nice 3D images of what the tail tube what the tail tube proteins look like. So yeah, I think I like images, and this paper has some quite cool ones.
Yeah, this was an interesting, definitely like the structural biology part of it. Sort of, like I wasn't like I didn't absorb too much of that, but they did focus on this idea that there are adaptations in this particular tails protein that help it slot together properly, and then they try to interrogate that through doing they do mutations, I believe, and then they then they're just looking at the tails and they do mutations to it, and they see like under different conditions how do those mutations change the dynamics of things coming together. Yeah, is that all you want to say about this one?
That's all I wanted to say about this one.
Yeah, that's all for me too. And I just wanted to then follow up then with this comment that Leo made in the chat, just asking about, like, in our last paper, we're talking about doing a forward bacterial screen, and the way that they did the readout for that screen was they deep sequenced like the whole library. But in the past, you could imagine the way that you would conduct that is by picking colonies and like Sanger sequencing each one. I think I'm not sure actually, because I haven't budgeted out like a big deep sequencing a library. But I would assume that it's like these days it's like at the same cost because you have to do all that Sanger sequencing. But the infrastructure cost would be higher because you have to have access to like those deep sequencing machines, and that's not like in everyone's wheelhouse. So like, for example, like at Biotech Without Borders, we could do a colony picking screen, but it might be a bit more difficult for us to do a deep sequencing screen depending on like where we have access to the core facility. Yeah.
Yeah. We can move on to the next paper, which is plasmids manipulate bacterial behavior through translational regulatory crosstalk.
Yeah, this is I thought this was a great paper that I saw. Actually, I found this one through the Fediverse. Yeah, which is, just another example to get on there. If you haven't been, there's a strong microbiology cohort there now. And this story starts with this idea of looking for so there's like, you can look at protein similarity and kind of guess that these are like the same types of proteins, similar function because they have similar sequences. And the types of proteins they're looking for are transcriptional regulators, stuff that is going to control gene expression. And so they're looking for transcriptional regulators on plasmids, and they find there are a bunch. So that's a really interesting observation in of itself in my mind, because it means that there are these proteins being manufactured by plasmids, which are mobile genetic elements that are at least predicted to have some sort of impact on gene expression, like the global gene expression profile in cells. You're K-Bus, you're like a...
I'm sorry, I'm just like coughing a lot. I'm trying to mute myself whenever I cough, but I'm not sure I'm doing a very good job of it.
Not all succeeding.
But
yeah, so right, it's like really kind of fascinating that some plasmids could probably modulate the global gene expression of cells, and so they go in and they test it, right, with this specific example of RSMQ that they identify on a plasmid that I think transmits between Pseudomonas species. And it's a pretty standard paper in the sense that they show that it binds to a set of cells, that they can do like they can do a large, I think, I'm not sure if they do mRNA-seq or do they do proteomics? Oh, they do both.
yeah.
Yeah, I think they might do both. Or at least they do proteomics is what I'm reading. And they find that, like, yes, expression of this RSMQ by the plasmid is changing a whole bunch of gene expression profiles on the bacteria. And specifically, it changes some phenotypes like its conjugation efficiency, the types of nutrients that it does better on. Yeah, just like some very interesting stuff that you would imagine like really changes the lifestyle of these Pseudomonas that take up the plasmid.
I mean, this is what is fascinating about me, because like it changes the biology to help promote its own like spread, which to me sounds like virus behavior. This is like the kind of behavior you'd expect from like a virus or a viroid or something like that, but it's not quite there yet. It's like on the cusp. It isn't like it's not entirely negative for the bacteria they travel within.
Yeah,
and
I mean, I think it's hard to the way that they started asking this question was they looked broadly amongst all these different plasmids. There's a huge diversity of plasmids that get into Pseudomonas, right? And some of them, and many of them, we would expect by the sequence similarity, have some ability like this to influence their hosts. So I would expect that there's probably classifications of plasmids, right? Ones that are more antagonistic to the way the bacteria wants to be, and some that aren't.
So yeah, this kind of paper makes me think about what the line is between, say, a plasmid and a virus. They're both like kind of selfish genetic elements, but then what makes them like different from each other? And that's fascinating for me. So I don't. So this is like a fascinating paper.
Yeah, and so at least in this, and sorry, just to go back, because like you were saying, like it's like they're not all pathogenic necessarily. I just want to be clear. In this example, having the plasmid with RSMQ versus having the plasmid without RSMQ, that the fitness is equivalent, right? At least in the environments they tested. So like this change in lifestyle, at least in the way that they tested it, doesn't seem to be affecting the fitness of these organisms. So it isn't like, this plasmid isn't a pathogen, I would say, but it could exist, right? I think that's like a very interesting curiosity. Yeah, the fact that it changes the swarming behaviors, right? Like we've talked about this before, that like bacteria live in biofilms, and biofilms are group living situations, and so changing the propensity for a bacteria to live in a group or to live alone, like that is very interesting. Like in terms of like, where does the plasmid then get to have experiences? Will it be in a group living scenario where it can conjugate with its neighbor cells, or is it going to be more alone or like seeking new niches so it could spread further?
Yeah.
Yeah, I'm not sure. I would say to Leo's question, I don't remember enough of this paper. Like, I skimmed this some time ago. I'm looking off of my highlighted notes on the Zotero. I don't remember if I had like a thought about where it would necessarily be better at living with this plasmid. But I invite you to look at it yourself, perhaps.
Yeah.
Do you have any impression there, Buzz?
On like what? So I get the. So yeah, I mean, it's hard for me to think about because what I'm thinking about in the situation where this is in the soil and they infect like soil bacteria. So maybe
Hmm.
this like help forces them to be more in close contact with, say, roots or other soil kind of components.
Hmm.
Um,
Hmm.
but beyond that, I'm not sure I can say for sure how what
Yeah,
situationally.
yeah, like it makes them more like soil is a low nutrient environment.
Hmm,
So being able to use nutrients better and move further away, it does sound like a survival thing. And maybe the fitness difference they see, the reason they don't see the fitness difference is because they're not interrogating the actual environment it's supposed to be in, right?
hmm,
Like maybe RSMQ does improve fitness, but just not on an agar plate. Maybe
right.
we would see that in this kind of like low nutrient environment, more like a spatially segregated environment, something like that. All
Okay.
right, let's move on.
All right, next paper. Recruiting unicellular algae for the mass production of nanostructured perovskites. So perovskites are an interesting kind of crystal structure that follow a specific kind of formula, and they're getting quite important for engineering. So they're minerals that apparently have applications in superconductivity, uh, like magneto resistance, telecommunications. So you see them often being talked about in terms of like LEDs, photovoltaics, and especially for solar panels. And so they basically have like a specific structure that I don't really want to go into too much, but it's like an ABX like 3 or something like that. It's
That's
so
like
the
a
original one,
crystal. That's a crystal morphology of some sort.
yeah. So yeah, and the interesting thing is like the properties of perovskites are dependent on their structure, and uh and there is a lot of like work into trying to like create like new perovskite structures. And this
Mm-hmm.
one was quite interesting to me because I've heard about perovskites because I used to work on an engineering journal not so long ago. So I know that they're quite important for like opto like electronics and other things like that. So
Uh-huh.
the idea of creating them using algae is very interesting because again, you're taking a photosynthetic organism and then using it to create some photovoltaics. There's some kind of like things are rhyming there.
Definitely.
So
Yeah, and from what I understood from the paper, this algae, like, it makes a shell. It's the shell that is this perovskite structure.
yeah,
Hmm.
so a lot of like algae like create shells. Like diatoms have like glass silicon shells.
Hmm.
This algae creates like a carbonate shell. Uh,
Hmm.
and what they do here is they take algae that have had this make this carbon shell, and then they kind of kill off the algae to get just the carbon shells, and they use some chemistry to exchange the molecules in those carbon shells for like the molecules for perovskites. So the idea is like they'd take the structure of the shell, which is like a calcium carbon like trio CaCO3, so calcium
Hmm.
carbonate, um, and then exchange the calcium for one metal and the carbon for another, and thus
Hmm.
create a perovskite out of that. Um,
Mm.
and so this is basically mostly a chemistry paper that has some microbiology attached to it. Um,
Well,
and
the microbiology was required to make the scaffolding, right?
yeah,
That they then have to do some chemistry to transform into something that is going to become the basis of a photovoltaic cell.
yeah, exactly. So I think the idea is that yeah, they've got these scaffolds created by these algae, and they use that to transform to create lead perchlorate. So that's
Mm.
PbCl2O8. No, sorry, that's using lead perchlorate to create
Yep.
the perovskite.
Lead, yeah, some sort of like lead carbonate, I guess, right? Like it's a
Yeah,
calcium carbonate that then goes into like a lead carbonate. That was what I was seeing.
yeah, that's
Um,
right. So
yeah, that's really interesting. I mean, the greater thematic element here that I think about is like in biotech tissue stuff, like they talk about like extracellular matrices and like making
mm,
scaffolds for cells to get onto so that you can get the right three-dimensional structure, because tissue is all about having a certain 3D structure that leads to a specific function.
Mm.
And it sounds like when I was reading this or skimming this, like what struck me is that like for solar cells, the 3D structure like that underlies the solar cell chemistry is really important.
Mm.
And these biological organisms happen to make like a mineral coat that has a great 3D structure. So it's addressing one of these issues of like how do you make such tiny complicated things? Well, I guess biology does it at some level, and then we can kind of co-opt that as a scaffold in order to make our desired our desired mineral shapes.
Yeah, I think that's it's kind of sets things out for the future. It's yeah, and there is potential for synthetic biology to create new scaffolding shapes that could in turn feed through to new perovskites. So I think that this is a very interesting paper in that kind of respect. So
Mm-hmm.
who knows
Mm-hmm.
where it will go from here?
I want to point out, like in the image of A, you can see like these shells that they make. I guess the flagella must come out of a hole at the back of them,
Yeah,
right? Like they all kind of look like little urns, and so I'm assuming
yeah.
that like that hole is somehow useful for for the biology of it all. Whether or not that has an impact on the ability for this to be a good photocell, I don't really know. I just wanted to point that out for
there is a
reading.
like vast variety of different like dinoflagellates out there that they could use. So
Mm-hmm.
there's plenty of scope for them to test with other kind of organisms to see what. Because it is fascinating. I mean,
I don't think to speak to Cordo's question, I don't think that the shape itself has like a benefit to the photosynthetic dinoflagellate. I think that this is its armor. I think it's an
yeah,
armor shape. Yeah, like this is supposed to protect the cell from getting crushed or like predated on in some ways.
hmm.
But who knows? I mean, like remember the minerals that the algae makes for itself in the shell, they aren't the same minerals that we're gonna want in this shape to
Hmm.
do the photochemistry. So yeah, but I'm not sure. It's unknown. There's an unknown part to that question.
Yeah,
Absolutely, which is what makes science so fascinating.
yeah.
All right, so let's move on. Next one we got is Cryo-EM structure of gas vesicles for buoyancy-controlled motility.
Mm.
So the first time I heard about these structures, gas vesicles, it was in the context of synthetic biology, which is wild to me because like the first time I heard about it, it's like it was like these gas vesicles they're made from a single protein is what I remember learning, and that you could induce their expression and then create bacteria that become opaque to ultrasound.
Oh.
Because like if you change their density, the ultrasound's going to look differently. And so the idea was that it could be like a marker for if you have to like look inside of something because the bacteria could have a switch that like they change their density based on some synthetic circuit. But this paper I think is a little bit more on the basic applied side, or not applied side, the basic biology side of it. They say, what's the structure of this anyways? How does it look in 3D? And maybe like as a theme of some papers that we often cover, what are the beautiful cryo-EM images of these particular structures look like? And so this structure appears in many different bacteria. They're going to look at the ones from Bacillus megatherium, I believe, because they're the simplest. Because some of them have like a bunch of other proteins that are important in the structure, but this one has like a nice simple one. They're able to make it heterologously in E. coli as well as purify it from the organism itself, and then using the magic of cryo-EM, meaning taking freezing it in a vitreous ice solution, taking lots of thin sections, looking lots of images, then using computers to reconstruct the three-dimensional image from all those 2D slices. They come to this just amazing structure of a single, well, actually, it ends up being two molecules that create cones. The kind of a helix of the single molecule rolls up and makes a little cone, and the two cones come together, and that is a gas vesicle. The inside of the cone is extremely hydrophobic, meaning that no water wants to get in there. And presumably there's a pore somewhere. I think they think it's at the tip, where gas molecules can diffuse inwards.
Yeah.
And that's the structure that allows, or that's what allows these things to get filled up with some sort of gas.
And the thing that I find quite fun is like how these vesicles are built up, because they kind of start off small, but they're built up from the middle outwards.
Mm.
So I've got the video cut up on screen where you like kind of have this small vesicle that's built outwards, and then as it grows bigger, it just builds out from the middle.
Mm-hmm. Yeah, that's a really cool video. But that's just done like that's not a molecular simulation. That's like this is like their hypothesis on like how it's growing, and they of course can see the snapshots, right? They can see snapshots of growth to verify their model, and they can see the seam. I think that's an important feature in all these images, right? The seam where these two cones come together, that looks a certain way in their EM images, and so like they have this knowledge. You can imagine that this is like not something easy to get a structure of because it's so big. So they have to be able to like stitch things together. But that's also the power of Cryo-EM these days, is that they're able to really zoom out and look at a lot of sort of from the very big to the very small. I mean, in this microscopic nanoscale even.
Yeah. Yeah, I think this is a great. I mean, I love the videos on this thing, and also just the concept of like dare I say, the design of this is really an incredible creation.
Yeah, good job, upper management of nature.
Good job, RNG function from evolution.
Yeah, I guess the last point that I want to bring up to the folks listening is that, as I said, there's a diversity of these gas vesicles in the bacterial kingdom, and the structure that they're showing us is really only one of them. But something that we've talked about before on the channel, and that's sort of a mainstay in structural biology, is once you have one structure, it can be easier to model subsequent structures. Because you end up having like a scaffold, and like you can start making like little predictions and like substitutions in, and like, you know, that's where computers, I mean, I don't know how it works, but like that's where computers take over. I think we talked about like they modeled like the entire structure of like a viral capsid or something at some point, right? And like that, they don't have direct structure from all of that, but like by combining multiple structures, they can figure things out. So what I want to say is that some of these gas vesicles have other proteins incorporated in them, right? And they have different substitutions, and so there's still like a lot of diversity potentially in the way that these gas vesicles are out there. Like maybe some of them grow faster, grow slower, grow under different conditions, respond to different concentrations of gas, maybe have different gases in them. I'm not sure. Right, but like, there's still a lot of interesting questions, I think, that this paper helps us like ask now, because we have a strong understanding of a basic structure of a gas vesicle. Yeah, Corridor says that the structures remind them of microtubules. For sure. Any sort of like automatically growing thing, I can see that.
Yeah.
What's this? I'm trying to think of the size. What's the size of a microtubule? Is it something?
I mean, I dare I say in a micrometer, or in that kind of level of is that, but yeah, I mean, the
microtubule diameter, because microtubules also have an inner section.
Yeah,
of course, it's not sealed off, but it's not sealed off.
Yeah.
Diameter. So these ones in Figure 1E, they give you the diameters of the different gas vesicles out there. So the one that they're looking at is the Megatherium one. They're looking at the smallest one. It's about 38 nanometers.
Right.
And a quick Google search of microtubules gives me the diameter of a microtubule being about 17 nanometers.
Oh,
so these are a lot bigger.
Yeah.
Or 25 nanometers. Yeah. These gas vesicles are bigger.
Right.
Not by much. Not really like a. Oh, actually, I mean, the largest gas vesicle estimating for us here are 100 nanometers across in diameter. So in some cases, these gas vessels can be an order of magnitude larger than a microtubule. But yeah, at the small end, there may be just like 2 or 3 times thicker. But from the self-assembly point of view, that's really interesting. And what that notion sparks in my imagination is microtubules have stuff that crawls on the surface of them, because like the repeating structure right, provides the place for myosin to attach. So I mean, I don't know, but like imagine what might be able to attach to the surface of this thing.
True.
All right, let's move on to the next one. We're a little bit over time, but let's see what we got.
Yeah, molecular fingerprints resolve affinities of the Rhynicher organic fossils. So, bacterial fossils. This is something that I never really think about, but apparently, like, there's this I guess slab of soil that was fossilized a couple of years ago. Well, I say a couple of years ago, millions of years ago. This is back from the Devonian period, which is like 410 million years ago. Apparently, this is like it's thought to be a set of terrestrial hot springs, and the soil somehow in this got fossilized, and it had quite a high. It fossilized to an extremely they can section off parts of it, and they look through it, and they found like funguses and potential bacteria in it. So this is bacterial fossils. So you see these pictures of like these fossils in there, and
I never thought about fossilized bacteria before, or just fossilized microbes, like because you imagine they're so small, and they always say like the structures when you think of bones being fossilized, they're like a lot harder than stuff that I think of in microbes, and they have to get replaced by minerals. But I guess in the right conditions, really strange stuff can occur.
Yeah, this makes me ask like how fossilization actually works, because like we think about like minerals being exchanged, so the structure of the organism, whatever organism or kind of organelle was present, kind of remains, but like what exactly happens there? And this paper actually tries to delve into the chemistry a little bit. So they use attenuated total reflectance FTIR microscopy. So I think it's Fourier transform infrared microspectroscopy. So this is usually a mechanism to find the chemical structure of things. So the great thing about it is it's non-destructive, and with fossils like this, you don't want to grind them down to figure out the chemistry. Having them underneath like a lamp, I say lamp, I mean infrared spectroscopy, a light source is much better. And so this paper,
although I'll interject and say that with all this DNA, all these ancient DNA studies, we are grinding down fossils. Larger and larger cohort of scientists that are grinding down fossils to get information out of them. But I'm sure, that like, there has to be good reason to. And having a non-destructive method allows you to revisit these in the future and maybe learn things from them. When you grind down like some mammoth teeth, you don't get those teeth back.
Yeah, exactly. You want to keep something around, and this paper kind of uses it. So through like just looking at the microscope, they kind of identify different species and say that some of them maybe look like algae, some of them look like fungi, and they use this FTIR to try and look at the chemical makeup of these fossils to see if they can distinguish them from each other. So what they did is they did like unsupervised analysis. So they have all these chemical traces, and they use some advanced data analysis to cluster them together to see whether like organisms are similar to each other, are similar chemically. So in this way, they can like classify.
Yeah, and that's driven by like our knowledge of like what molecules are in the living representatives of these species, right? That we know that like living bacteria have peptidoglycan, which means that it's going to have this sugar. Therefore, inside of the fossilization process, that could be driving some of the difference in spectra that we see. So yeah, I think it's really interesting to have to work backwards from that idea. But I think this is true in a lot of paleontology, right? Like you kind of have this understanding of how life is now, and then you're trying to say what evidence of it can we see in the past, and how might it be different based on the fact that we're in some ways looking at the rock replicas of of these previously living objects.
Yeah. So this essentially tries to like clarify like what the rock replicas of these molecules look like, so that can we work back from those. So for instance, while the Rhynie chert is very well preserved, there are other fossils that maybe aren't as very well characterized. And the question is could we use this data to perhaps learn things about other data? So if we find, say, another like fossil preserve that we can't like identify the organisms as clearly, can we use the traces we get from FTIR to work out what chemicals were there and maybe classify whether what we're looking at there is eukaryotic or bacterial? And I think
Totally.
I think that's can be very interesting, especially if you go back far enough back to where the differences between eukaryote and prokaryote maybe weren't as set out as they were now.
Oh my gosh, yeah, that that would be really wild if we learn more stuff about like Luca and things like this, right? By directly observing chemicals in fossils.
Yeah, I'd be very interested to see like whether they can actually work back from like the fossils to the chemistry of before, and maybe they probably won't get a perfect picture, but getting an idea of like, oh, we had these molecular structures that have been kind of bruised by fossilization, but maybe we could work back to figure out what was going on there.
Right. The level of resolution that they have with this technique is like it seems like from what I was reading, it's on the scale of macromolecules, right? It's like talking about like the big components. So it's like, are they made more of protein? Are they made more of lipids? Are they made more of carbohydrates? Yeah. So like, yeah, I mean, I don't know, like how those details could somehow influence the way that we think about evolution and picking up certain things to be adapted.
Yeah, I mean, there's so much potential there, but yeah, I do think that for small molecules, I mean, we're not being able to find out the DNA of these organisms from these traces. I don't think.
Yeah, and we're not going to get like for example, like we looked at structural biology last. We're not going to get that level of resolution from this either, right? Like because we're looking at rocks. These are rocks.
Yeah.
All of the biology has been replaced by the process of fossilization. And so like it's like one removed from that. But yeah, at the same time, like I look at these Rhynie chert micrograms and I'm like, there's a lot of detail here that like I didn't expect would exist at this scale, the micrometer scale. So yeah, I had no idea this field existed, and it's super cool to have a paper from it.
Yeah.
All right. Last paper that we have for you today is called Alcobiosis, an algal fungal association on the threshold of lichenization. I think that title is just hilarious, mainly just because like I didn't know that there was lichenization. I didn't know you could be on the threshold of lichenization. But for those who don't know, and you know, for those who also just want to revel in the idea of lichen, these are symbiotic pairings of an algae and a fungi. In some ways, they're so closely they're one organism, right? They're so closely in symbiosis that they can't live without the other. They're obligates. But of course, you can imagine that is like the sort of the most close evolved relationship that you could possibly get between two separate things. And there might be a gradation, a spectrum of closeness that you could see sort of between fungi and algae, right? Moving all the way up. And specifically, they focus on this idea that like some fungal hyphae like are a little bit more translucent. So like you could imagine that these fungi could just grow over an algal sheet, and then that would give them some close relationship because light could still go through them because they're translucent. It could photosynthesis, and the stuff underneath could, you know, share nutrients with the fungi, and maybe the fungi like protect the algae underneath from being predators or something like that. And so they look for examples of this. They go into the woods and they look at rocks and tree bark, and they pick up some really beautiful images of mats of algae that are co-living with mats of fungal hyphae. These people are just very. I think they are I think these people are taxonomists, and they just, by visual identification, they're able to say like, oh, this is this species and this is this species, and they're able to start talking about very specific. And I can't relay any of this information to you guys because I'm not familiar with these species names. But yeah, they're able to relay some information about like what are the pairings that they see in these environments. They even do like a crude form of stable isotope probing where they have a field device that's essentially a bell jar that they place over these sites, and then they're able to measure respiration out of the bell jar. And they can take gas samples, and they can say like, oh yeah, like we think that these algae are transmitting metabolites to the fungi above. And I think they do one more thing that I'm not sure if there's a picture of, but they look at snail grazing. and they find that like the snails grazing on the surface produce, oh yeah, it's this figure 7, right? Like the fungi have this like, dense kind of, they get opaque eventually, but snails grazing over it eat up the fungi, and then the algae are still alive underneath, and they're able to get a little bit more visibility to the sun. And so, yeah, through these lines of evidence, the authors tell a story of sort of these different pairwise observations that they made of an algae living in close relationship with a fungi. It's not lichen. It's not. They're not lichen because they don't need. They're not like a single organism. They're not like obligate. They don't have an obligate relationship with each other. But yeah, it's pretty darn close. It's a pretty darn close living situation.
Yeah, they're not in a relationship. They're friends with benefits at this point. They can exist on their own, and
yeah. Yeah, and I just thought that was really interesting, and so I wanted to share with you those papers. Yeah. But I mean, I feel like we're a YouTube channel. We're driven a little bit by images, but we're also both infectious disease microbiologists, and that drives a lot of our interest area as well.
Yeah.
So
speaking of like and subscribe.
Join us next. We're going to be back in 2 weeks, so we'll be off April. Hopefully, Fuzz, you'll be better. Thanks for soldiering it out today to run this. Yeah, we'll be back in 2 weeks for more cool microbiology papers.
Yeah, and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
Yes, I agree. You can reach out to us over Twitter or on Mastodon. We both believe that peer review is a process, and that it requires folks to actually have read the papers to make it work. So if you think you have something to add or you found something unclear, just let us know. And I do just want to take a pause before we say goodbye. Finally, someone asked, are there any animals that have photosynthetic microbiomes? I believe we covered a paper in the past about slugs that eat algae, and they sort of maintain photosynthetic microbiomes. So yes, the answer is yes to that. I couldn't type it fast enough.
Yeah, I mean, it's been a pleasure chatting with you, Danny, and thank you to all the commenters who have been really active this time around. It's so good to hear from people.
Yeah, it's really nice to have folks to chat with and sort of geek out over some of the papers that we found. So yeah, tune in 2 weeks for more microbiology content.
Yeah, see you then. Goodbye.
Bye.